Okay, so here's a very quick guide to getting started with Adapt. I've got a data set here, 2 channels, time-lapse, movie of a cell. One of the signals is totally sinusolic, and the other is a signal that we're interested in quantifying. So we go to Plugins, Adapt, Analyze Movie, and it'll initially ask you where you want to save your outputs. So I'm just going to select the desktop, and then it asks you which of your channels is the cytosolic one and which one is the signal of interest to be analyzed. So you make sure that they're selected appropriately. Click OK, and you get presented with this. So you can go ahead and click on Preview down here, which will generate a segmentation. That may or may not look appropriate for your purposes. We'll talk about how you can adjust that in just a second. But I'm going to run through the settings here as they're presented. I won't go through everything in the interest of time, but starting with the simple tab, make sure that the spatial resolution and the frames per minute are appropriate, and in my case I know that is incorrect, so I'm going to change that to 12 because movies of this are required every 5 minutes. Most of these settings are pretty self-explanatory, but you can find more details in the wiki on GitHub if you need more information. All of these buttons here concern the outputs that get generated. I'm going to leave all of them selected for now. Now I'll move on to the Advanced tab. If you find that the segmentation that Adapt is generating is not very good, or it's not satisfactory, so for example if you see something like this, so you can see that the default thresholding method is not working terribly well in this particular case, you can select one of the others. So you'll find here the list of all of the thresholding methods that are built into Fiji. I found in my case here that the Huang method works fairly well, so I'm going to go with that. You can change this, the image gets smoothed before this segmentation occurs. You can change that if you want, so you should end up with if you increase it, you should end up with a smoother cell boundary. I'm going to put that back to 1. A couple of other things that might be of interest: the cortex depth. So that defines the width of the region of interest that's defined around the cell boundary. You may want to either make that wider or narrower depending on your needs. You can change the thickness of those lines in the visualization as well if you want to, if they're not clear. That's obviously a bit too thick, so I'm going to put that back to what it was. That looks absolutely fine. That's just for the purposes of this preview. And finally, we've got some filters here that get applied to the velocity maps and the signal maps that get generated in the output. So when you look at your outputs, I'll give an example when this is finished running. You may find it appropriate to alter these. They smooth the data in the output maps basically, so you may find it necessary to filter, to increase or decrease these values depending on your particular experiment. In this case, just to highlight the protrusion analysis tab, I am not analyzing individual protrusions in this case, but you can find more details of that in the wiki GitHub. So that's about it for this particular data set. So I'm going to click on Run, and it will generate a bunch of stuff in the log window as it goes through each frame and segments the cell, and then it will analyze all of that data, build cell maps, quantify fluorescence, localization intensities, and it will dump a whole load of data in this output folder up here that's been created on my desktop. So I'm just going to wait for it to finish, and then we'll go in there and see what's been generated in just a moment. Almost done. And obviously how long this takes will depend on how big your data set is and how many cells you have to analyze. Okay, so that's all done. So if I now open this, we'll find that there is a subfolder for that particular data set that was just analyzed, and you'll find various different folders in there. You'll also find a file, an XML file, that contains all of the settings that we use to generate this data, which is handy for reference purposes. And there's different folders containing different things. So you've got individual cell output. We'll have a look at that in a minute, but the first thing we're going to look at is the visualizations output, because that's the first thing you're probably going to want to check to make sure that the segmentations and the tracking actually worked appropriately. So in there you'll have a couple of different different folders which are essentially just image sequences. So you can load each of those image sequences in Fiji by going to import image sequence, and then point Fiji to that folder. Where are we here? Visualizations. I'm going to look at the velocity visualization, and this might take a minute to load again depending on how big the files are. But this is a good means of verifying that the cells have been both segmented appropriately and tracked appropriately. If that hasn't worked properly, then the rest of the data probably won't make a whole lot of sense. So it's usually a good place to start to make sure that this looks approximately correct. Okay, just another moment. So this will load a 32-bit image that contains a representation of the cell boundary, and along each point on the cell boundary is an approximation of the velocity of that point on the cell boundary at each point in time. So you're probably going to want to select an appropriate lookup table to visualize this. I recommend the ICA ones are quite useful for this. And then change the brightness and contrast to an appropriate value. So you set the minimum and maximum to some approximately what you would expect the minimum and maximum velocities to be in your data set. I've arbitrarily chosen -10 and +10, but I can see that that's worked quite nicely in this case. So in orange we can see regions where the cell boundary is expanding, and in blue where it's retracting. Okay, so that looks reasonably good, and you can overlay that onto the original data if you want to, just to be absolutely sure that the segmentation is reasonably accurate, but that looks pretty good to me. Okay, so that's a reasonably sure sign that everything is run as it should. But just to show you another couple of outputs very quickly before we finish up. Going back into that results folder, I mentioned the individual cell data output, and you'll find a bunch of different things in here for each cell that was in your movie. I'm just going to show you, for example, the velocity map, which basically shows you the same data that you'll see in that velocity visualization, but this time it's been flattened out into map form. So again, we can apply the same lookup table to this, the ICA one, and again set similar brightness and contrast limits, like so. And here we can see if we moving along an X, you're basically moving forward in time, and moving up and down on Y shows you different points on the cell perimeter. So this shows you basically all of the regions in your cell that are expanding and retracting as you move through time. Okay, and you can do things like, for example, add a calibration bar to that. So if you go to Analyze and Tools, you can add a calibration bar, which will appear in the top right of your map if you like. Okay, so that's a very brief introduction and overview of ADAPT, and you'll find the test data that I've used in this movie on the in the GitHub repo, so you can try all of this yourself. And you'll also find this example output results, so you can verify that everything is working correctly for you. Okay, so I hope this was helpful. Please do get in touch if you have any other questions.